Hello dear students, welcome back again to Chenmoy's Biology channel. So in the previous episode we have discussed about the basis of the fundamental unit of life, that is cell. So today we are going to discuss in details about the plasma membrane, about the nucleus and what all are present in the cytoplasm of the cell. So let's get started because it's a very important lesson. Stay tuned and don't skip the lesson in between because I'll be teaching the important thing in the middle as well as till the end. So stay tuned till the end of this lesson. So let's get started. So in the previous episode we have discussed about the basics and we have discussed that what is the structural organization of a cell. So there we have discussed that the main three components of every cell, what should be present there is the plasma membrane, the nucleus and the cytoplasm, right? So today in details we are going to discuss about the plasma membrane, nucleus and cytoplasm. So let's start with plasma membrane. So what is this? What is the plasma membrane? So this is the outermost covering of the cell which is separating the contents of the cell from the external environment. So it is the outermost covering, as you people are covering your classwork, notebook and all like that only. It is the outermost covering of the cell which is separating the inner content of the cell with the external environment. Now the plasma membrane, what does it do? It allows or permits the entry or the exit of the materials in and out of the cell. So some of the materials which are inside the cell needs to be excreted out of the cell, so that is they should move out of the cell. So that in and out of the cell is always controlled by the plasma membrane. So it also prevents the movement of some other materials also. So the cell membrane is called a selectively permeable membrane because it is not allowing each and everything to enter or get out or move out of the cell. So it is selectively permeable. So what does this selectively permeable mean? This means that it selects and allows some material to go inside the cell, and then like that only some material can move out of the cell. It's not like that full throughout whatever is there outside will move inside and whatever is inside can move outside. No, it's a selectively permeable membrane. Now, how does the movement of substances take place in the cell and how do they move out of the cell? So this is what we should know. So listen very carefully. So some substances like carbon dioxide or oxygen, they can move across the cell membrane by a process called diffusion. Right? So what is diffusion? The process of diffusion in earlier chapters we have already discussed, right? So diffusion is when some substance from a region of higher concentration is moving to a region where there is lower concentration, right? So we have seen that diffusion is a spontaneous movement of a substance from a region of higher concentration to a region of lower concentration, right? So this is diffusion, and this is what happens when some substances like carbon dioxide and oxygen are moving across the cell membrane, right? So similarly, this happens in cells when, for example, we have taken some substances like carbon dioxide, which is a cellular waste, and they requires to be excreted out by the cell, accumulates at high concentrations inside the cell. So when too much carbon dioxide is accumulated inside the cell as a cellular waste, so it should be moved out of the cell. So how it is happening? As there is a concentration difference, because carbon dioxide is more inside the cell and it is less outside the cell. So like that, by the process of diffusion only from the cell, it moves out of the cell. Now the concentration of carbon dioxide is low as compared to that inside the cell. So as soon as there is a difference in concentration, it moves inside and outside, from inside to outside of the cell. So like this, carbon dioxide moves out of the cell. So it's very easy, right? When the concentration is higher inside the cell, it will move from inside to outside. When the concentration of something is more outside the cell, it will move from outside to inside. Right? Is it clear to everyone? Now, similarly, oxygen enters the cell by the process of diffusion. When the level or the concentration of oxygen inside the cell decreases, thus the diffusion plays an important role in gaseous exchange between the cells as well as the external environment. Okay, so water, we can say that water also obeys the law of diffusion. The movement of water molecules through such a selectively permeable membrane is called osmosis. So what is diffusion? It is the diffusion is the movement of certain substances, and diffusion of water through the semi-permeable, selectively permeable membrane is the osmosis. Now we will move on to that in details in the next slide. Now we will be discussing about the movement of water through the plasma membrane, right? The movement of water across the plasma membrane is also affected by the amount of substance dissolved in water, right? So the water is having some substances dissolved in it, so it is dependent on the substance also dissolved in water. Now, thus the osmosis is a net diffusion of water across the selectively permeable membrane towards a high soluble concentration. So, water is moving along with the solutes in it from a region of lower concentration to a region of higher concentration. So what will happen if we put an animal cell or a plant cell in a solution of sugar or salt water? So this observation we should know that what is happening when we are putting an animal cell in sugar or salt solution or a plant cell in sugar or salt solution. Now, one of the following three things could happen. First, if the medium of the surrounding cell has a higher water concentration than the cell itself, so meaning that the outside solution is very dilute, the cell will gain water by osmosis. So such a solution is known as hypotonic solution. So the medium surrounding the cell has a higher water concentration, that is, it is more dilute, so water is moving from outside to inside. So the cell will gain water by osmosis, and the solution here is the hypertonic solution. So the solution here is the hypertonic solution. Okay, so now we will move on to the next one. So the next one is when the water molecules are, now here what is the condition? The water molecules are free to pass across the cell membrane in both the directions, but more water will come into the cell than will leave. So the molecules of water are free to move inside or outside, but more water will come into the cell, inside the cell, than to leave the cell, right? So the net result is the water enters the cell and cell will swell up. So there we will see the constant swelling of the cell as water is moving by osmosis from outside to inside of the cell. Now the second example is if the medium is exactly the same water concentration as the cell, so what will happen then? So then there will be no net movement of water, so it is having the same concentration. So the cell and the water is having the same concentration, so there is no movement of water inside and outside the cell. So this is called an isotonic solution. So water crosses the cell membrane in both the direction, but the amount is in the same as the amount going out. So there is no overall movement of water. The cell will stay the same size. So whatever water is moving inside, that amount of water is only moving outside. So the cell volume, the cell size will remain constant. Okay. Now the third condition, what is it? So first one was hypotonic solution. The second one was isotonic solution, so same concentration inside and outside the cell. And the third one is if the medium has a lower concentration of water than the cell. So if the cell plant or the animal cell is having higher concentration than outside the cell, that is the medium is having lower concentration of water, that is concentration of water is lower in the outside, that is it means the solution is very concentrated. Okay, so the first one was solution was dilute, so it was a hypotonic solution. Second one was having equal concentration, so it was a isotonic solution. And now the water content of the outside part of the cell is less, that is it is more concentrated. That means that it is very concentrated and the cell will lose water by osmosis. Such a solution is known as hypertonic solution. So three kinds of solution: hypotonic, isotonic and hypertonic solution, right? Now again water crosses the cell membrane in both directions, but this time water leaves the cell more than it enters, therefore the cell will shrink. So the cell is shrinking in this case, right? So hypotonic, isotonic and hypertonic solution. So in the third case, the cell will shrink, and the osmosis is a special case of diffusion through a selectively permeable membrane. And now let us try out this following activity, right? We will try out this activity here. Now the first activity is osmosis with egg. So this is activity 5.3. So please do this activities at home so that you can understand the process of osmosis diffusion through a selectively permeable membrane. So what we need to do, remove the shell of an egg by dissolving it in dilute hydrochloric acid. So this all ingredients you will not be getting at home. So you need to do this experiments or activities in school only, but with the help of a teacher. Now the shell is mostly calcium carbonate. A thin outer skin now encloses the egg. Put this egg in pure water and observe after 5 minutes. What do we observe? The egg swells because water passes into it by osmosis. So you are just dissolving the outer part with dilute hydrochloric acid. And the shell is mostly made up of calcium carbonate, and the outer skin is now enclosing the egg inside, and it is put in the pure water. And now we will see that after 5 minutes the cell size has increased. So it is water is passing from the outside to inside. Now place a similar de-shelled egg in concentrated salt solution and observe after 5 minutes. So what will happen in this case, as the concentrated solution is outside, the egg will shrink. And why? Because the water passes out of the egg solution into the salt solution, because the salt solution is more concentrated. Is it clear to everyone? Now we are clear with these two experiments that what is osmosis, what happens when the cell swells up, and what happens when the cell shrinks. So another activity, similar activity is there. We can also, this type of activity we can try with raisins and apricots. So if you are soaking raisins at night, in the morning you will see the cell swells up. So water from the outside, normal water you are putting from the outside, will flow inside because the water concentration is less and the solution is hypotonic. So water will enter the raisins and the raisins will swell up. Now same with the apricots, as this we have told it is a case of hypotonic. Now we are putting another this activity. See, put dried raisins or apricots in a plain water and leave them for some time. Then after some time what we will observe, that each gains water and swells when placed in water. However, whenever you are putting them in concentrated solution, it loses water and they consequently shrink. Okay, so this is what is happening. Now in case of animals, what is happening, we will see this also. This is very important. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis, right? Now absorption of water by plant roots is also an example of osmosis. Now thus diffusion is important in exchange of gases and water in the life of a cell. So in a life of a cell, diffusion is very important, and in addition to this, the cell also obtains nutrition from the environment through this process of diffusion, because different molecules move in and out of the cell through a type of transport which is requiring the use of energy, right? So this energy, what is this energy here? This is ATP. So moving in and outside the cell through a type of transport which is requiring the use of energy, right? Now the plasma membrane is very flexible and it made up of organic molecules called lipids and proteins. So what is the plasma membrane made up of? It is mainly made up of lipids and proteins, right? So we can observe the structure of the plasma membrane only through an electron microscope. It is very flexible. It is made up of lipids and proteins, but we can observe it through an electron microscope only, right? Now the flexibility of the cell membrane, what is its advantages? So advantages is it enables the cell to engulf the food. Now when we were studying the food and digestion, we will be studying food and digestion in amoeba, we will study in the 10th standard that how it is making the cell only as it falls, feeds and engulfing the food outside it. So amoeba is a unicellular animal. It is having a cell membrane. So cell membrane only, it's acting as its feet, and it's holding the food and it's engulfing it. So they are used as engulfing the food also in other materials from the external environment. So such as the process is known as endocytosis. So I'll show you the process in the next slide with the help of a diagram. You just see how that happens. And amoeba acquires its food through such processes only. That only I'll teach you in the next slide. Now here you see, find out about the electron microscope from the resources in school library or through internets and discuss it with your teachers. So what is an electron microscope? How does it look like? Now you can go to the internet and just search it. Now there are few questions here to be discussed about. So how do substances like CO2 and water move in and out of the cell? Just now I have taught you each and everything in this class itself. So just take your answer book and write answer the following questions. So why is plasma membrane called a selectively permeable membrane? That also I have discussed in this lesson only. Now we will see how the amoeba is using its plasma membrane to engulf the food in the next slide, and then we will move on to the cell wall and the nucleus, right? Now, how the plasma membrane is modified to engulf food in amoeba, in this diagram we will see that. Just see here, this is the plasma membrane only. So how it is modifying and folding the plasma membrane and using it as its pseudopodia. So pseudopodia is its false feed. So see, this is the food particle. It is spreading its plasma membrane to form false feeds and then engulfing the food inside that, and then gradually it is engulfed inside. Amoeba grows, the food is absorbed inside, and the undigested food material is thrown out of its body. So this is the full process of digestion, assimilation and digestion in amoeba. This no need to know in your 9th standard, but in 10th you are having this in details. Just for your knowledge, for your extra knowledge, I am teaching you that how this the external cell membrane is being modified as the false feed. Now we will move on to this nucleus and the cytoplasm. Now we are moving to cell wall. So cell wall, what is it? Cell wall, the plant cells, in addition to the plasma membrane, they are having another rigid wall, is called the cell wall. So this cell wall is not present in animal cells, it is only present in plant cells, right? The cell wall lies outside the plasma membrane, and the plant cell wall is mainly composed of cellulose. So what is the composition of cell membrane? We told that cell membrane is made up of lipids and proteins. So just make it clear when you are studying each part, it should be clear in your mind. And your basics, if they are clear, it will be very easy to answer your questions. Now cellulose is a complex structure and provides structural strength to plants. So cellulose is very strong. It is providing the structural strength to plants. So when a living plant cell loses water through osmosis, there is a shrinkage or contraction of the contents of cell away from the cell wall, right? So inside the cell, plant cell, the cell membrane shrinks, so it moves away from the cell wall. This phenomena is called plasmolysis. Now, if you are not understanding this plasmolysis, shrinking, diffusion, hypotonic solutions and all, go to my lessons. I'll be providing the link in the description box as well as in the link below, where I have taken each and every topic separately and discussed. So go to that part and learn this topic separately. Plasmolysis, we can say that hypotonic solution, isotonic solution, everything has been discussed in detail. So here no need to discuss in details, just we are telling that this process of contraction of the cell contents away from the cell wall is called plasmolysis. Now we will take an activity here, 5.6 activities. Let us discuss this activity. Mount a peel of rio leaf in water on a slide and examine the cells under the high power of a microscope. So what do we note? Now we have seen that small granules are called chloroplasts are present in the leaf. They contain a green substance called chlorophyll. Put a strong solution of sugar or salt on the mounted leaf on the slide. So on the slide itself you are putting the solution. So what will happen? We are waiting for 1 minute to observe. What do we see? Now place some rio leaves in boiling water for a few minutes. This skill, we can say that this kills the cells. Now then mount one leaf on one slide and observe under the microscope, but put a strong solution of sugar on the mounted leaf on the slide. Wait for a minute and then observe again. So what do we find? Did plasmolysis occur here now? So we will see the observations. Here what we did, we have we were having chloroplast cells, chlorophylls, and there in the chloroplast cell, and then on that we have put the strong sugar and salt solution. Now we have boiled these leaves and we have mounted, because these boiling of the leaves, they are killing the cells, and then we are mounting on the slide, and then we are putting the strong solution. So what is the observation now? So what do we infer from this activity? We are inferring that it appears that only living cells and not the dead cells are capable of absorbing water by osmosis, right? So cell walls, they permits the cell of plants, fungi and bacteria to withstand very hypotonic external medium without bursting in its cells, such as it tends to take up water by osmosis. The cell swells up, building up a pressure against the cell wall. So cells are swelling up, so they are building a pressure against the cell wall. The wall exerts equal pressure against the swollen cell. Because their walls, such cells can withstand much greater changes in the surrounding than animal cells, as they are having a protective layer of cell walls outside the cell membrane. As the cell walls, they are permitting the entry of from the cell walls also, the water is entering the cell. The cells are swelling also a lot, but they are able to resist. They are not exploding or they are not bursting out, right? Cell walls are present there, so they are not bursting out. So these are the differences rather from the plasma cell membrane and the cell wall, if there is cell wall present. If there is no cell wall present, the cell, if swollen to a large extent, they'll just burst out. But in case of plant cell, they'll not burst out. So this is all today till cell wall, and with activities I've discussed today. The nucleus part, cytoplasm and other cell organelles I'll take in the next class, because today only I have made it vast, so I don't want to make it vast. So stay tuned, stay updated with all the lessons which I am posting, and please do subscribe to my channel for the upcoming lessons which will be more interesting, and I'll be teaching your lessons as per your curriculum and in an easy and systematic manner. So stay tuned, stay updated with all my lessons. Thank you.